Welcome, everybody. My teacher voice. If you could hear me clap once. Look. No, thank you. Welcome back. And now I have the pleasure of introducing Dr. Simon Heeke. He has spoken to us before on our Zoom call, and he is the assistant professor for the University of Texas at MD Anderson Cancer Center. So, can I get the slides? Okay, perfect. Thank you so much. I'm Simon. It was really nice meeting many of you, and I'm really excited to share a little bit our research on biomarker development. I should say, biomarker development and small cell is usually, not the thing that usually, attracts a lot of attention because there are not so many biomarkers. So after having all the questions that came in yesterday, I actually had changed the slides yesterday evening because I felt like, hey, we can show more. So I'm really, really excited about this because I'm also the head of the Liquid Biopsy Translational Working Group. And usually I spend a lot of time explaining to people what are liquid biopsies, how we can use them. But again, after 7 questions yesterday, 14 questions today, Misty's talk, I feel like, you're all ready, to really dig deeper into it. So I'm really, really excited. Before I start speaking about small cell, and I think this is also the story that always came up, let's first have a look at non-small cell. Because I think when we now look at the drug approvals in non-small cell lung cancer, just in 2022 and 2021, and frankly, I haven't yet done, updated the slide for 2023, but I'm pretty sure it looks pretty much the same. So when we just look at the drug approvals in non-small cell lung cancer, you see like every month almost something happens. There are times where within a single week, we see the approval of 2 new drugs in non-small cell lung cancer. And I think this is really, really where we want to go with small cell lung cancer. I want to show the same slide for small cell lung cancer. But the reality is, unfortunately, that when I have to start speaking about small cell lung cancer, we go from a scale of every month, we go over decades. And the reality is, for many, many times, really not a lot of things happen. We didn't saw a lot of new approvals, and the really new big thing that happened was just, a few years ago when we started to see the approval of immunotherapy, usually in a combination with chemotherapy, that started to gain accelerated or full approval in small cell lung cancer. But as nice as this shows, the reality is that even today, we have already lost approval for 2 of those regimens because the data didn't hold up. So as much of a hype as we really want to generate and going into new drugs for small cell lung cancer, the reality is that we are very, very far away of the situation that we have in non-small cell lung cancer. And one of the reasons, or there are many, many reasons related to it, but one of the reasons I believe is really that what we are lacking a little bit in small cell lung cancer were biomarkers. And if you remember what Christian Lovely was saying yesterday, so biomarkers are generally something that tells us if a therapy works or works not. So that's the general idea that we have about biomarkers. And it spans a lot of different things that we have. And I'm going to highlight a little bit more of the complex biomarkers that we are developing in small cell lung cancer, hoping that this might really be the avenue that we need to go into those, good therapies. The important thing, and I want to start with something positive while I spend the rest of my talk probably highlighting a little bit more the, challenges that we face, especially the challenges that we have when we move something from a research setting to the clinical setting. Because the important thing is I'm a PhD by training. I don't see patients, I don't treat patients. I create biomarker strategies for our clinicians. So when I'm speaking here about something that we can do right now in a research setting, this has nothing to do with what our medical oncologists can really do in routine clinical care. So please be kind to them and please be patient to them and don't run to your doctor but Simon said we can do this. The important thing, however, is we are working on it, and they are allowed to send me angry emails. So to start with something positive, and I think this is something that I really want to highlight, and that's something that you've seen yesterday already, is there are biomarker-driven clinical trials in small cell lung cancer. So that's the positive thing that I wanted to start with. And probably the one that brought the most attention recently is the SWOG 1929 trial. I think Choparna Sen and I think Christine Lovely also spoke about this yesterday. And not speaking too much about the biological idea of the clinical trial. The important thing is, it was a randomization to treat patients with a PARP inhibitor versus, patients only treated with atezolizumab, which is an immunotherapy. The important thing I wanted to highlight is really here in red, that this was a clinical trial that included a biomarker called schlafen-11. And patients were randomized whether this protein, which we call schlafen-11, was detected in a protocol that we call IHC or immunohistochemistry, where we take, tumor slides and then we can stain it. And if there's the protein, we see the color of the slide changing. That's the positive part. The probably more challenging and honest part is that IHC is something that we do since decades. And it's something that, even putting a slide on, speaking, positively and excited about something new in any other cancer type would be a little bit, people would look at you like, hey, we do this since the 90s, we do this since the 2000s. So the positive thing is that we see biomarker-driven clinical trial. The negative thing is we still have a very, very long way to go. And I hope I can share with you a little bit why I believe that this is. And one thing, and if you ever want to know how you can assure that research you did is good, is when you create a paper where one figure is shown in, I think, every single presentation that we had in the past today. So I think you're so familiar with it that, even know it by heart. So Julie George and, a large team did a fantastic job in really showcasing the genomic composition about small cell lung cancer. And we spoke about this. The important thing is, what we usually and typically see is that we see mutations in two genes that we call TP53 and RB1. And importantly, we do not see something that we call actionable mutations. We call them actionable because, we have a drug targeting towards this. This is something when we speak about EGFR, when we speak about ALK, when we speak about RET, and all those, new fusions or new genes that come up in non-small cell lung cancer. Those actionable mutations are something that I wouldn't call it easy, but it's something that has drawn a lot of attention and was used in clinical trials and is now used in clinical care, because to a certain extent, detecting those mutations is technologically relatively straightforward. The most important reason for this is that it's a yes or no. You have a mutation or you do not have a mutation. There can be technological reasons why we do not detect mutations, but the important thing is when you detect a specific EGFR mutation, you know that this mutation is in the tumor, and when you have a drug, that you have a certain chance that this drug might work or not. So it's a very binary outcome, and this is very important because at the end of the day, when you think about when you treat cancer, when you treat a patient, you can do it or you cannot do it. So there is, to a certain extent, a gray zone, but at the end of the day, this binary outcome is important. The situation that we have in small cell lung cancer with the absence of those actionable mutations is that we go in a much, much more complex situation that you've also heard before. Because small cell lung cancer is not defined by the mutations that we have, like when we speak about EGFR mutant non-small cell lung cancer or ALK fusion positive non-small cell lung cancer. But small cell lung cancer, or the classification of small cell lung cancer, has really evolved over decades in an increasingly complicated scheme where we separated in a neuroendocrine and a non-neuroendocrine subtype. And within these neuroendocrine subtypes, also something you've heard now a lot, we call it small cell A, N, Y, or I will tell you, why we call it the small cell I and the small cell P subtype. So that's like the idea that we have. We're not speaking about mutations, we're speaking about those more profound, really characteristics of this cancer, whatever this means. So just to add, what we actually did is that we were highlighting an unbiased approach to small cell lung cancer. Actually, those are my old slides, I think, but that's fine. About small cell lung cancer subtypes, and we actually came up with the same thing. We highlighted this a few years ago, where we really saw that small cell lung cancer is driven by the expression of three, we call them transcription factors. I will speak about this in a minute, or those three genes that we call ASCL1, NEOD1, PAU203, which you've probably heard now a couple of times. And therefore, we classify now small cell A, N, and P. And in addition to this, what we detected was something that we call an inflamed phenotype. So this is, a type of small cell lung cancer where we have a lot of inflammatory cells in there that we call the small cell I. But admittedly, those classification, there's a little bit of a debate. It's still a little bit fluid to a certain extent. So the important thing is just remember that we have those, different types of small cell lung cancer defined by those 3 genes that are really, showcasing or to a certain extent highlighting biology that we have there. So now the question is, how can we develop biomarkers from small cell lung cancer subtypes? So how can we use this and develop something new? And the important thing is that we really go away from those mutations where we say you have the mutation or you don't have the mutation to a much more subtle differences that we have there, because we really see much more profound phenotypical differences that we see there. And I think one of the reasons that we have not yet seen those biomarkers where we want them in the clinic, we want them to be used, at least, starting with clinical trials, but obviously, that every patient with small cell lung cancer gets them. And one of the reasons why we are not yet there is simply because doing something like this, to the best of my knowledge, has never been tried or approached. Definitely not in lung cancer, but also in other cancer types. Because really, the backbone is, to a certain extent, we have the expression of certain genes. Can we target those genes? We have the presence of a certain mutation or the absence of a certain mutation to then decide for a treatment. But something like a very, even what you've heard, heterogeneous, but very subtle differences in, the cancer type that we have there is something where, to the best of my knowledge, we just have never tried it. And this is a lot of the challenges that we have faced in order to really bring this to the clinic. So I'm really trying this. Be patient with us. We really give our best. But I will also tell you at the very end why I think this is, at least worth working on. The important thing is now when we try to develop biomarkers for those complex states is that we are generally faced with a little bit obvious probably a problem. And the problem is that the more information we get and the more information we integrate to really get the best biomarker that we have and get the best characterization of the tumor, the more we are increasing technical complexity. Because as always, the more information you bring in, the more of a chance you have that the information is either biased or that the information is wrong or there's something happening just technologically that we don't control. And this is really a very, very important problem when we now speak about small cell lung cancer. To highlight this, I will show you now a little bit, a stage where we are currently and to really see why we have to work on this. Because what you see here, so this was presented at a conference 2 years ago. This is transcriptional data from the Caspian trial, which which was a very fundamental phase 3 clinical trial leading to the approval of immunotherapy in small cell lung cancer. And what we were looking at together with other teams is now how can we understand, those small cell lung cancer subtypes in this clinical trial. And the first and probably, obvious approach was like, we know that we have those genes that you've heard about, ACL1, PAU203. Why don't we just look at those genes, how they are expressed, assuming, the highest expressing gene is the winner. So when you have more of one gene over the other, then we call it that subtype, versus when we have more of another gene, then it will be another subtype. The alternative contrasting approach was the approach that we did to, better understand and a little bit develop those subtypes was that we took 1,300 genes. So we took the complete other extreme of taking many, many genes and really trying to understand how the increase of information can really balance subtyping. And what you see is that when we just look at single genes, that's not really going to work. We see like there's an absolute over-representation of the small cell A subtype. It's not really reflecting what we've seen previously, and why there is a lot of, good data in this and really helped us to better understand how we use small cell lung cancer subtypes, it still doesn't seem to be the right approach to just look at one gene. On the other side, when we now look at 1,300 genes, we see relatively balanced differences in our subtypes, really reflecting what we've known previously from cell lines and from other data for those subtypes. But the important thing is that working with this vast amount of information is also a problem, because working with too much information is really something that dramatically challenges our way of bringing those biomarkers really robust in the clinic. This is something in a research environment, this might work. But to really bring it to the clinics, this needs to work with thousands of different samples. This needs to work in different institutions, different countries, different conditions. So while that looks very, very good, and it's nice to show to you, the issue is still that probably this is too complex. And we really need to find a way that we can maintain the biological information while reducing the technological complexity that we have there. So one of the reasons, or one of the thoughts that we can now have is, how can we actually achieve this? And now it's the important part to speak about those transcription factors. Because those three genes, ACL1, NERD1, and PAU2F3, are transcription factors, meaning that those are genes that orchestrate the transcription of many, many other genes. So while we think that we look at one gene when we look at them, actually we look at really strong differences in many, many different biological pathways that are really orchestrating the phenotype of small cell lung cancer in general. So this goes to the idea that when we now look at a conductor and think about those transcription factors as a conductor, obviously you can look and say like, okay, this is a very classically dressed conductor. Maybe this is a conductor conducting a, very classical orchestra. And here I should say I made this with JetGPT, so let's see how good it got it. But the idea is here now we have a little bit more in a 20s style dressed conductor who is orchestrating a jazz orchestra. And here, this was supposed to be a conductor, orchestrating a musical theater. And when you look at this, you already start to be like, but it can also be this, or it can also be that. So to a certain extent, when we look at the conductor, we can get a little bit of an impression of what is going on there. But it's not perfect. And this is something that we see because there's too much complexity below there. So the better idea, or the interesting idea, is what if we don't look necessarily at the conductor, but we look at the orchestra? Because when we now look at the orchestra, when you look and you see like 20 strings and, 10 celli and everything you have there, then you have a pretty high probability that this is probably a classical orchestra. Well, when you see, you see a lot of basses, you see whatever you have there, trumpets, and everything that you would expect in a, jazz orchestra, then you might have a good chance, hey, that's probably a jazz orchestra. Of course, even there is a certain heterogeneity. And when you have this, pop appearance that you have there, hey, probably that's a musical theater. And that's a little bit the idea that we are trying to bring in, that why we have those vast networks that are regulated by those transcription factors, we not only focus on those on the conductor itself, but we are really trying to understand how are the networks that are regulated by them different, and use this to develop biomarkers. And I think this already gives you the first glimpse why it's really much more complex than what we're usually used to doing with like biomarker development. And the important thing, and this is really very, very important, because this is the big step from going from a research-based biomarker into something that we can use in the clinic. And this is that we really, really need to take what we have and simplify it. So for example, I'm from Germany, and in Germany I will teach you now, we call this a Dackel. I think you have a different name here. But I think the important thing is when you look at the dog, everybody of you, I think, know this dog. How you call it is not important. You all know, and how do you know it? Because the dog has certain features. It has shorter legs, it has this long back, it has a certain, face. And I think it's pretty easy for all of us to understand which dog breed this is. So now think about what are the features of this dog and how can we make it much, much easier and simplify it in the best way possible. And when you're a great artist, which I'm definitely not, but when you're Picasso, then you can now see that with a single stroke, he actually perfectly manages to take all the features that you have there. And I'm absolutely sure that all of you would agree that this dog or this sketch is this dog. And this is what we are trying to achieve. And the important thing is when we take it too far, because you're not a good painter like I am, so this is my one, Then when we now take the simplification too far, what is going to happen is that we actually lose too much information. Because, maybe in the context that we have there, you would say, yeah, okay, to a certain extent, Simon, you got it in the right direction. But now think about taking this completely out of context. And I only show you this figure. You would ask a lot of questions, and you would probably question a lot of things that I've done in my life to reach to the point that I claim that this is this dog. So the whole beauty of bringing those biomarkers to the clinics is actually, and I think this is the perfect way of phrasing it, that from going very, very complex, we have to find the best simplicity to this point where we can no longer reduce the complexity that we have in our biomarker development. And what you see here is actually the real challenge that we have, and this is what takes time. Thinking about those, small cell lung cancer subtypes is one thing. But the reason why you hear a lot of this, you hear a lot about the biology, but you have not yet seen this in the clinics, is that this is the process which is really, really challenging and takes a lot of time. So with this as a basis, usually I now start to speak about machine learning, because machine learning is the cool thing where you say, make me a figure, and then you end up with, 4 legs, 19 fingers. And I've learned something about speaking about machine learning. So when I present, the way we use this methodology to really achieve this simplification, which is very important there. And when I speak to my colleagues, what has usually happened that, most of them sleep or not interested, or get a backslash to when they were 12 and had math lessons, or, had to do math with their parents. So it's not the most appealing thing to talk about, admittedly, even with my colleague within research. So I was a little bit like, how can I present this here? And I recently learned the other extreme, which was I was speaking to a patient who was really interested in the research, and I realized that the patient was an engineer and actually knew much, much more about machine learning than I did. So I have now a 50% chance of either boring you because you don't want to hear about machine learning, or boring you because you're like, hey, whatever you're saying there is not right. Let me tell you about machine learning. So for the sake of this conference and for all of us to not embarrass myself and you're not getting bored, for the rest of the talk, when I speak about machine learning, then just imagine this is this magical thing that happens and then gives us the new biomarker that we need to work on. And as stupid as it sounds, it's a little bit like this. So having now, reduced how we did it to the absolute minimum, I will quickly guide you a little bit how we are trying, or how we're currently working on really bringing now those biomarkers into development. And what we were actually doing, so we had a very large cohort of small cell lung cancer samples, and we were now using 2 different methodologies, and then using we called it predictive models. So this is all the machine learning part, and it's actually all the reduction that we have there to create something that we can use to do the subtyping, and I'm happy to give you more details during lunch break when you have an hour or 2 and tell you a little bit more how we actually achieve this. And the important part is we use 2 different methodologies. The first methodology is something that we call RNA expression. So RNA expression is the way our genes are created. And this is a, so to say, direct way of looking at what we do there, because as I highlighted before, we look at a lot of different genes which are giving us the phenotype of the small cell lung cancer subtypes. And when we just look at this and we see that RNA of gene X is at 10 and RNA of gene Y is at 20, then we would argue, okay, this is directly correlated to certain biological features, and thus we can now use it for small cell lung cancer subtyping. The other methodology we use, and you've heard already in the talk before, is DNA methylation. I don't want to go too much into the details, but DNA methylation is a way how our DNA is changed, which allows us to have a certain indirect way of understanding gene expression. So we don't look at how much of gene X RNA is there, but it's also a very robust way to understand gene expression because of the biological principles that we have there. So just keep in mind we have this RNA expression, which is direct, and we have this DNA methylation, which is the indirect way. And we now use this and build those, predictive models for like subtyping, and really try to bring those, subtyping approaches into, more clinical setting. And what you see here, and I promise you this is the most busy and crazy slide that I will show today. You don't need to completely understand it, no worries. But the important thing is that we were now looking at a cohort that we have published recently with 142 patient samples that we had, going back to discussions what we do with, when we get samples. We were really profiling them. And we also had a second cohort of another 207 patients. And we used our new fancy methodology, all the magic and the machine learning. And what we could demonstrate, and I think this is the important part down here, is that first of all, we managed to get a meaningful result for 96% of all the patients. So this is important because, as I said, the important idea is to make it robust. All biomarkers have sometimes flaws. Not everything works 100%, and this is something that will persist and that will not change. But at least we really managed to use this method for the vast majority of the specimens that we had available, and this was very, very reproducible. The other thing which is important to look at, which goes back at what we had in the beginning, is how are those subtypes distributed? Because we know that there's a certain biology below, so we would expect that when we look at a certain group of patients that X amount of patients will have a tumor which is small cell A, Y amount of patients will have a tumor which is small cell N, and so on. And what we saw and what was really encouraging for us, and this is the distribution that you see down here, is that when we used our new method, that the distribution was very, very homogeneous in the sense of independently of which cohort we look at, we always came up to a certain extent to the same distribution of the small cell lung cancer subtypes. And we also had the pleasure to look at a couple of other data sets that we have there, really highlighting that what we see there and what we classify is likely represented by the biology. It is reproducible in the sense of we use different cohorts, different studies, and we really get there. And this means that we are really going the right way of creating those biomarkers and bring it into the clinics. The next thing then was, and I'm really, really excited, I'm so sorry because this is where I was actually updating our slides a little bit, but I will still guide you through, is speaking about liquid biopsies. Usually I plan to spend a little bit of time to speak what are liquid biopsies, but I know you're all aware about this. So let's make it brief. The idea of liquid biopsies is that we take not a tumor and try to infer information of the cancer, but that we take a blood draw, and in the blood we find certain material derived from the blood cells that we can then use to infer information on what is going on in the cancer. And obviously, there are many, many advantages. It is used especially in non-small cell lung cancer, but it's particularly interesting in small cell lung cancer. And among other reasons, the main reason for this is that when we now look at different tumor types, and we look at something that we call ctDNA or circulating tumor DNA, which is, so to say, the amount of DNA that we have in the bloodstream that is derived from the cancer versus the DNA that is just, from blood cells, from all the other biological processes that happen in your body, then we see that small cell lung cancer is actually the cancer type of all solid tumors where we have the highest amount of information. And this is very, very important because usually liquid biopsies are really challenging to work with because we really have to work with very minute amount of tumor-derived material. There's a lot of what we call background, meaning that we have DNA from blood cells, from liver cells, from all the other cells in your body, because when they die, they release, their material in the bloodstream. So usually it's technologically a very, very challenging field, and it's really something where we need a long time to really understand how to do it. And small cell, we're now in the situation that actually we have so much of information from the tumor that we have a very, very good chance to develop something that really allows us to use blood instead of the primary tumor sample. And the important thing is always when you do something, and there are two possibilities. Either you're the only one doing it, this means either you had the first idea or it's not a good idea, or you're doing something what a lot of people are doing. And in the case of small cell lung cancer, it's exactly this. So the idea of using liquid biopsies, and what you've just heard from Misty before, is that it's something everybody is interested in. And everybody is interested in because we all know how powerful it can be in small cell lung cancer. And those are just a couple of scientific publications that I wanted to highlight, which came out really just during the past 2 years, 2 or 3 years. You know, like Joe Hyatt from Fred Hutch published like last week a very interesting paper on how to use liquid biopsies for the subtyping approaches. And you can really see, you know, I don't need to go too much into the details of the methodology, but it's like everybody in small cell lung cancer who has an idea of using liquid biopsies is working on it. And this is important and this is reassuring because, you know, if you would be the only person, you sometimes, you know, might just not be on the right track. But here we really see that this is something we need to work on. And I have to say, some of the most brilliant new methodologies that have been developed in liquid biopsies in general, in my personal opinion, have been developed specifically for small cell lung cancer in the past years. And this is very important, and I will speak about this a little bit at the very end, is because from this cancer type, without real biomarkers. Why do we need biomarkers? There is nothing. Small cell lung cancer seems to be now one of the leading cancers for liquid biopsies in general. So when you have a new technology, when you see new companies coming out with new technologies, they are now all very actively working in small cell lung cancer. So from this, oh, we don't want to work with small cell lung cancer, we really see the field changing to, oh my God, I definitely want to work with small cell lung cancer because this is where we can really demonstrate how good our methodologies are. So that's the positive part, but keep in mind, research to clinical implementation takes some time. In my updated slides, I now added a slide actually that came up about small cell lung cancer detection, which I also saw there were yesterday a lot of questions around this idea. And the important part is that those liquid biopsies, as you might have heard, I think there was a lot of press, you know, coverage everywhere, is that the liquid biopsies in general have been increasingly developed with the idea of cancer detection. Christine Lovely yesterday spoke about this, what we call the MCEDs or the MCED, the multi-cancer early detection test. And this is something which is currently under development. Again, this is in a research field. The important thing is some of those assays that have been developed for lung cancer in generally have shown a sensitivity. So the probability that when you have a cancer that is detected of 70 to 80%. Meaning they seem to be very, very good in lung cancer in general. And the positive thing is, when we now look a little bit at the smaller data, and as we've heard earlier, small cell lung cancer is actually relatively easy detectable. I say relatively easy compared to other cancer types. Not like, you know, there's still very complex technology used. Meaning that there might be a chance to really detect small cell lung cancer in the future from those small cell lung cancer subtypes. We heard 12 out of 13, Misty? 12 out of 13 were detected with small cell lung cancer. So you really see that we might slowly be going in this direction. The problem, and this is again what takes a little bit of time, validating those assays, confirming that you can use this, takes a lot of time. And usually how this is done is in studies with, you know, healthy individuals in the sense of cancer-free individuals, which are then followed over time with a blood draw. And then over time, when, you know, the study participants develop cancer, then you can say like, okay, we detected it or we did not detect this in the sample. Now, what this highlights to you is that we need gigantic cohorts of probants to really work on this. So the studies which are currently ongoing have tens of thousands of people participating in this. So it really takes a little bit of time ongoing there, and small cell lung cancer, which is still a relatively rare cancer, you know, might just need more time. But again, we are going in the right direction. It's not yet there in the clinical setting, but we are really actively researching this in the research setting. So the question is now, why am I speaking so much about liquid biopsies? Because this DNA methylation that we've talked about yesterday, today, this indirect way of detecting gene expression in small cell lung cancer is actually preserved in our liquid biopsies. So whatever we do in the tumor, to a certain extent, at least we have a chance. I'm just saying we have a chance to do this also in the liquid biopsy. And now comes the other busy slide, and now I will guide you a little bit through this to better understand this, because this is what we actually did in our study. So for the patients we had 142 where we get this RNA expression data. In total, we had 179. And for, I think, 80 patients, we also had a corresponding blood draw, which then allowed us to really compare how this is working. And what you're seeing here is actually how we develop those models. And I will guide you through this. So we created something that we call consensus classification, which for the technical detail is not really important for the moment. But what is important is that the color that you see there corresponds to a certain amount to the probability that a sample is positive for one of those 4 small cell lung cancer subtypes. With the more green you see that, the higher the probability. And what you also see up here is the small cell lung cancer subtype, with each of the subtypes having a different color. And then we call this our GRC approach, which is just our direct RNA expression methodology from tissue. And now our liquid biopsy approach that we call CFDMC. Not important what this stands for. What is important is what you can see here is that when we now look at our data, that the subtype that we assigned based on our tissue-based classification system is the same subtype that we assign based on our liquid biopsy assay. So meaning that what we do from tissue can also be done from liquid biopsy with a high probability. We really have an almost perfect overlap there, and really showcasing that we can now use this liquid biopsy approach in a research setting also to better characterize and look at small cell lung cancer. And this is very important for multiple reasons. The first reason is we spoke about diagnosis. So obviously, how nice would it be that you can just give a blood draw, you know if you have small cell lung cancer or not, and if you have small cell lung cancer, what's your subtype, that in the future, when we might have specific therapies for this, you can get all this information quickly from a blood draw. The other thing that is very important is that, as we've heard, small cell lung cancer is very heterogeneous. And small cell lung cancer changes, and small cell lung cancer changes when it's treated. So the important thing about small cell lung cancer, and I think this is why the longitudinal liquid biopsy collection is so amazing, is that with liquid biopsy, we have the opportunity to follow how the tumor changes over time, that we can really be, hopefully one day, ahead of the curve to really adjust our treatment based on how the tumor changes. Again, I'm speaking here strictly in a research context. And actually what we saw when we now looked at our cohort is that we also had some patients where we had another liquid biopsy at progression to treatment. And the important thing is, now that you got a little bit used to seeing this very easy and understandable figure, obviously, is that when we now looked at the classification at baseline, what you can see here, so we had most of them had the small cell A, few of those patients had those small cell I. When we now looked at progression, that many of those patients changed their subtype. And this is something that is probably to be expected because of this heterogeneity. This is the, so to say, bad part of it. But the positive part is that with liquid biopsy, we are now capable of detecting this, following this, and seeing how the tumor changes to really try one day be so much ahead of the curve that we can really, work and adjust our treatment based on what we have in a liquid biopsy. And to give you an example here, this is, data from 2 patients that we had. And one patient, when we looked at the liquid biopsy, the small cell lung cancer of this patient was a small cell A. And when we now looked at during treatment time, we always saw that this is really, a small cell A. And it changed, it didn't change. When we now looked at the second patient, and already at baseline, so the patient had already a little bit of treatment when it came to our clinic, we saw already a little bit of heterogeneity. Keep in mind, the color corresponds a little bit to the probability of the subtype. So we see that this patient was a small cell, or the tumor of the patient was a small cell A, but we already saw that there was something going on with like, hey, probably it's small cell N, something is happening there. And then during treatment, we saw something that we call the equivocal state. Because during the treatment, we saw that the tumor changed so much that we couldn't even assign a subtype because it became so heterogeneous that we couldn't even say it is, small cell A or small cell N. And this is very interesting because this was shortly before the clinical progression of this patient. And then we saw at progression that the tumor in this patient completely switched from a small cell A to a small cell N. And this is very important because this really highlights to you how liquid biopsy can track this, how liquid biopsies can give us not only a baseline information on the small cell lung cancer subtypes, but also during progression. And I think this is to a certain extent beautiful because most of the approaches we have seen is that we had a treatment, then we had the development of resistance mechanisms, then we had assay developed for this resistance mechanism, and we were always a little bit behind. So now that we can already demonstrate that we see it even before we start, treating specifically distinct small cell lung cancer subtypes, I hope that we might be a little bit ahead of the curve to at least, already have the technology to then, when something happens, we can follow. And to end with this and give a little bit of an outlook and, the differences of how we can now bring it to the clinics, I want to highlight something that was presented by Ann Chang from Yale earlier this year at the TTLC conference in Santa Monica. And this is a proposed biomarker clinical trial. To the best of my knowledge, this has not yet been approved, but I want to just give you a highlight how now clinical trial design can really be used for the development of novel clinical trials. And the idea, again, this is not yet approved to the best of my knowledge, is that we can now really use those subtypes that we have there. We've heard about Schlafen-11 and really integrate this to have clearly biomarker-driven small cell lung cancer clinical trials. Again, this is the future, but I hope I could show you a little bit how we can really, how we really hope to develop something like this in the future and not only, showcase you some fancy slides and be here and speak about, but really enable bringing this to the clinics. Keep in mind, this is the future. We hope it gets started. It might be different as I'm showing it here to you, but really, I think we're going in the right direction of those biomarker-driven small cell lung cancer. So in summary, I think this is something that have heard a couple of times, that small cell lung cancer really consists of the transcriptionally defined subtypes, that small cell lung cancer subtypes hopefully one day might guide precision medicine in small cell lung cancer. We really want to go to personalize the treatment that we have there. You have probably now been a little bit convinced that in a research context, small cell lung cancer subtype can be detected in the blood to enable this tracking of tumor evolution over time. But still, and this is very important to highlight, that biomarker development in small cell lung cancer is still ongoing. There are many, many teams working on it. And we are really hoping, that however this will look like, we have multiple biomarkers in the future. And before I go on the acknowledgement slide, there's one thing that I want to highlight that I mentioned in the beginning is small cell lung cancer was not the tumor we studied biomarkers in. It was not there. We don't have biomarkers. All the fancy work we've seen was mostly non-small cell lung cancer. But what is now happening was, now that we learned to understand those transcriptionally defined subtypes that we see in small cell lung cancer, we started to look in other cancer types as well. And for example, just last week, I was presenting some research that we did where we actually used the technologies and the idea that we had built specifically for small cell lung cancer, now in EGFR-driven non-small cell lung cancer. So despite the fact that small cell lung cancer was not the tumor where we had all those biomarker development, we're now slowly going in a situation where actually small cell lung cancer is leading the way of novel biomarker development, and that small cell lung cancer is now the poster child for the biomarker development and other tumor types as well. Again, I'm speaking in a research context, but I think this is really exciting and important to highlight that, there is change. We are working on it. It might take a little bit of time, but we are really working on getting novel and really complicated biomarkers in the clinical trials and hopefully in the clinic. And with this, so the important thing is that there are a lot of people involved in this. So all of the research was led by John Heymach and Lauren Byers. I started this when I was a postdoc, somewhere here in the back. We have many, many collaborators, and the important part also, which came up now during all the discussions is, and I'm speaking here as an employee of the MD Anderson Cancer Center, which is, I think, the largest cancer center in the world, or at least in the United States. We had a lot of partners to make this possible. we had collaborators. We were working with, for example, Christine Lovely, who you've met yesterday. We had collaborators from Spain, from France, from Austria. We are working with teams in Korea. We are really working internationally all together because only then we can make this happen. So this is the important message also, because there were so many questions: how do we work? How do we use and leverage tissue? The answer to this is together. Because only together we can really achieve this and really get the information that we need. And it is really important also to highlight that we have many other people now at the MD Anderson working on other types of small cell lung cancer. For example, Bingnang Zhang was recently highlighting how small cell lung cancer that is transformed from non-small cell lung cancer is looking to a certain extent differently. And we also have Kai Concannon, who's working on small cell lung cancer in never or in light smokers. So we are really trying to better understand small cell lung cancer as a general, but it's also very important at the very last to really, we need money to make this research happen, and we have a lot of funding agencies. We are very grateful, but nothing of this would have been possible with all our patients, with all of your support for allowing us to use the material, for supporting it, to coming in even when there's probably not the clinical need, for supporting our research, spreading the word. So really at the very end, I need to thank all of you really for helping us, that we can help you. Thank you.
Thank you so much. We are running a little behind. Lunch is waiting, so let's go ahead and take one or two questions. I know there's probably going to be a lot of questions for Dr. Heeke. That was fantastic.
Thank you.
Who would like to have? Okay, Wendy? Anyone else? We'll go ahead and get you lined up.
Thank you. My question for you, this actually is very timely because it came up in the Facebook support group last week, where somebody had said that their doctor was using a blood test as well as scans to determine as if their cancer had come back. And the specific blood test that they referenced was the NSE blood test. Do you have any information on that? They said it was a neuron-specific NLase blood test.
Actually, I don't have specific information to that test, but it's encouraging to hear that there are a lot of, development in this space.
Yes.
Thank you. No worries.
I can speak to that question. The NSE has been a biomarker for decades, like probably 30, 40 years, that similar to like chromogranin or synaptophysin. It was a blood-based test, neuron-specific enolase that is a potential surrogate. It's not perfect. So we see this also with like CA19-9 or CA125. Some of these tumor-specific biomarkers that are blood-based can be insightful, but still non-specific. So I would take it with a grain of salt. Yeah.
The important thing to probably add, however, is the second part, the integration with radiomics, because this is something that we are also actively working on. Again, this is research in progress, and we see that, obviously when you combine, again, I spoke a lot about simplification, and now, we blow it up again on the other complexity. But we have also seen that when we now add this radiomics information, a lot of other information, that we can really improve, the accuracy of our diagnostic tests. So this is really research in progress, and it's good to hear that it is really something that is used.
Simon, love your passion, love your presentation, love your energy, and thanks for helping explain what machine learning really is. But when you consider the work that Misty's doing in the longitudinal studies, AI is getting kind of a bad rap in some instances these days, but could you maybe explain how machine learning combined with AI might actually make a difference with some of those studies in terms of predicting the future?
Okay, but this will be a longer than 1 minute answer.
Well, just okay. Maybe say props up for AI.
No, I think this is an important part because it allows me to clarify a few things. So the first thing which is important is that machine learning is nothing new. So for example, there are algorithms, for example, we call this logistic regression models, which are very often used. They have been developed at the end of the 19th century. So we have, to a certain extent, machine learning models for a long time. The important thing is usually when we speak about machine learning, there are multiple methodologies. And one of those methodologies that we hear a lot are called neural networks. So this is something that is used in a lot of those really fancy and advanced machine learning methodologies, like with ChatGPT. And those are complicated models in the sense that you build really, really gigantic networks. The idea is to rebuild how our brain works. And to a certain extent, they're a little bit of a black box. So you feed data in, and it's sometimes really, really challenging to understand what is happening there. So engineers are working actively on understanding what is there, but it's a very, very complex way of doing it. It is extremely powerful, but it's very complex. The way how we approach this machine learning idea is that we work with methodologies that are much, much more, I wouldn't call it easy, but that are much, much more reproducible, where we can always control what is happening. And this is the big, big difference. So we don't end up with like 7 fingers or 8 fingers, but we use models that do not have usually the possibility to make those, hallucinations, well, I don't even know how to pronounce it, generating new data out of nothing. So that's a little bit the idea that we have there. So it is very distinct. No, that's the basic principle. So AI, or machine learning, artificial intelligence in general, is very important. Outside of ChatGPT, the reality is, and probably this is something to add, AI is used everywhere. When you go and have a radiomic scan and they make the scan, the images that you get are so nice and so crisp and look so wonderful because there are already machine learning and artificial intelligence algorithms working in it. So it is already there. We already use it. It's not just when you get your phone and you get a fancy photo. So it is there. We still have issues like 7 fingers, 4 legs, and some racist chatbots that are out there. But it's a very big field, and there are ways how, to a certain extent, you can control it. And this goes back to the simplification, because the simpler the model, the better it is. And this is why we use models where we can really work on this. And it's probably very long before lunch break. I'm sorry.
No, thank you so much. Thank you so much.